In Chapter 5, we're going to look at exploring genes and genomes. So basically, we're focusing on what do we do with the DNA information, how do we manipulate it, how do we study it. Now, I will point out that at a couple of places in here, for a couple of the techniques, you'll see hyperlinks for videos that have a little bit more detail about that particular technique. And so I'll also post those to the course shell so that you have easier access to those supplemental videos in addition to this lecture. So just to outline what we're looking at here, we're going to talk about recombinant DNA technology, look at key techniques for gene exploration, talk about recombinant DNA, vectors and their implications. We'll talk about genomic libraries, and it's not shown here, but we'll also talk about types of mutations. So there are a number of advances in biochemistry that have allowed for the advancement of recombinant DNA technology. And what we have listed here are some key areas that have led to the advancement in recombinant DNA technology. So first, we have the use of restriction enzymes. Now, most, if not all of you, have used a restriction enzyme at some point in the lab. So the use of restriction enzymes allows us to fragment DNA, and then those fragments can be manipulated. Hybridization with complementary DNA or RNA. DNA using cDNA or RNA probes will allow us to detect specific nucleotide sequences using some more advanced technologies. We are able to form recombinant DNA. So we need to have the technology to insert foreign DNA into host organisms. We have methods for determining nucleotide sequencing. So there are some different techniques we can use in order to sequence our DNA. I'm sorry, I've already said this one, our ability to deliver foreign DNA into host organisms. So we're creating vectors that are going to allow us to make DNA. So all of these areas together have allowed for advances, not just in biochemistry, but also in areas like agriculture and medicine. So with the key techniques for gene exploration, these are techniques that are key in exploring genes and that have led to rapid progress that's been observed in the biotechnology field. And we're going to explore each of these in more detail, but just to list here, we have restriction enzyme analysis, blotting techniques, DNA sequencing, solid phase synthesis of nucleic acids, and polymerase chain reaction or PCR. So looking at restriction enzymes, remember, are molecular scalpels. So these are going to cut portions of our DNA. They're also referred to as restriction endonucleases, so enzymes that we're going to cut. These enzymes function by recognizing a specific base sequence within the DNA, and then it cleaves both strands at a particular point. And that's kind of what we're seeing here. You have the recognition of a particular sequence and then cleavage of both strands of DNA to cut this. So different restriction enzymes are going to recognize different sequences and cut in different places. Typically, the point that they recognize is going to be a 4 to 8 nucleotide sequence, and that sequence is palindromic. Okay, so 4 to 8 nucleotide palindromic sequence will be recognized by the restriction enzymes. Now, many restriction enzymes have been isolated, so several hundred exist. They've been isolated, purified, characterized, and these enzymes are going to be named for the host organism, followed by the strain designation for that particular substance. So if we're looking here, this HAE3, for example, this came from Haemophilus aegyptius, and so the HAE comes from the host organism, and the Roman numeral 3 indicates that it's strain 3 of that particular enzyme. Now, the restriction fragments are going to be produced by this, and they're produced by the action of that restriction enzyme. The fragments can be cut by subsequent restriction enzymes, and that will allow us to produce even smaller fragments. And the fragments produced when we have restriction enzymes acting on our substance or our DNA, that will serve as the DNA fingerprint. So we're creating a DNA fingerprint through this digestion with restriction enzymes. So once you break down your DNA into smaller fragments, what do you do with it? Well, you run DNA electrophoresis. So remember that the phosphodiester backbone of DNA is negatively charged. That negative charge helps stabilize and protect the DNA backbone from nucleophilic attack. It keeps it together. So because of that charge, we can use gel electrophoresis to separate the DNA fragments. In general, the shorter DNA fragments are going to migrate further in the gels. So we have shorter down here. And the longer fragments are going to end up by the top. Now in DNA electrophoresis, we're using polyacrylamide gels. Or agarose gels could be used. And they're used to separate the fragments, and they separate based on size. So the content of the agarose that's used within these gels, that can be varied, and we vary that depending on the size of the fragments that you're trying to separate out. And then once you run this, the information can be transferred or preserved using one of several different blotting techniques that we have. So basically, blotting techniques, what we're doing is we're taking our gel and we're transferring that information onto blotting paper or nitrocellulose sheet. And that way we have a copy of what we're looking at. And we can do some other things with that. So one method that's focused on here is something called southern blotting. And southern blotting is a technique that is used to transfer DNA and label it with a DNA probe for identification. And we're trying to identify a particular fragment within that DNA. Okay, so southern blotting is going to be used for DNA. Okay, and we're transferring that DNA. So you take your sample, you transfer the DNA by the blotting process in the overall southern blotting technique. Then you add some sort of label that recognizes a particular portion, and then you have some additional tests here, autoradiography, which you are going to be able to detect that substance. So the gel is transferred to nitrocellulose sheet. Position of the DNA fragments are preserved during that transfer process. That nitrocellulose is exposed to a label. Here it's that it's radio labeled or an isotope, radioactive isotope. It could also be a fluorescent probe that's used there. The probe hydrolyzes with the fragment of DNA that has the complementary sequence to it. So it allows us to look at a particular portion. And then some type of imaging can be used to identify the position of that particular fragment of interest. Now there are other blotting techniques that you may have encountered or heard of. We also have Western blot. So Western blotting is another technique which use the same general process. However, Western blotting is used for protein analysis. And usually Western blotting is involving some sort of use of antibodies and secondary antibodies. And then we also have Northern blotting. And Northern blotting we are going to use when we're trying to study or look at RNA analysis. So Southern blotting is for DNA, Western is for proteins, and Northern blotting will be for RNA. So we do have a method for sequencing our DNA, for figuring out what the actual sequence is, and I think that is easier viewed through a subsequent video. So the link is here, and again I'll post this on the course shell so that you can more easily access this link. This just takes and shows you a visual of the process of sequencing. But we do have a figure and a slide that looks at how we're going to go through the sequencing steps. So when we're using this Sanger method to sequence, we have our dideoxy analogs of those base pairs, so the ATP, the CTP, the GTP, and the TTP. This is what the dideoxy analog portion looks like, and then the base, of course, adenine, thymine, guanine, or cytosine, depending on what's there. And each of these dideoxy analogs is going to be labeled with a different fluorescent probe, and the wavelength of that particular probe, or the color of that particular probe, is going to vary depending on which one it is. So basically, as you go through the process, you have your, we're looking up here at the top, you have your template strand, and from that template strand you add a primer in here, and onto this primer you're going to start growing the chain based on the template strand. So these base pairs, which are fluorescently labeled, are going to be added one at a time to the growing chain. Now each time one is added, you are going to see a band that corresponds to the fluorescence of that particular dideoxy analog. And based on, we're going to have these run through a column. It's capillary electrophoresis that we're using, but they run through this column, and based on the time that they come out, they're going to separate, and the color that comes out, we're able to put together that sequence. So this is a step-by-step process. We're adding one base pair at a time, or one base rather at a time, to that template growing primer that's based on that template strand, and we're able to get the overall sequence out. Another technique is solid phase synthesis. So in solid phase synthesis, we are going through sequential addition of activated monomers to a growing chain. So an activated monomer is just a reactive form of the piece that we want to put on there. So you have your bead. Usually these are done on some sort of bead or solid support. So this resin here represents our solid support. And on that solid support, you have some sort of portion that's present onto which we've provided some functionality onto which the growing chain is going to be added. So the monomers come in, and the monomers at first are going to be protected so that side reactions can't occur. So they come in, and we're going to have reactivity that occurs. You're adding this, and you end up growing that chain. Once you grow that chain, then you have to, we have iodine, which is, the iodine is going to allow for oxidation. So you see the change in the structure here. Once we've added that on so that we're creating that negative charge and stabilizing that backbone, once we've added this on, this portion here, this DMT, that was what was protecting this so side reactions didn't occur. Then we add another substance, dichloroacetic acid, to deprotect that or remove that. So we end up with a hydroxyl group that can then react with the next activated monomer. And we repeat this step over and over again until we get the chain of desired length. So this figure here, we're just looking at that DMT, or that particular, here's our DMT, that's that protecting group that minimizes side reactions. So it blocks reactive groups from getting in and interfering with this process of being added to the growing chain. Now, another technique we use is polymerase chain reaction, or PCR. So depending on the courses that you've taken here, you may have used PCR. We do have a PCR instrument. Generally speaking here, primers are added to a solution that contains a target sequence. Okay. And the primers will hybridize to areas that are called flanking sequences. All four of our deoxyribonucleic acids, or all four of our DNTPs, are going to be added into our container, as well as a heat-stable DNA polymerase. So you have to have a target sequence, you have to have primers, You have to have all 4 of the D NTPs, so DATP, DGTP, DCTP, and GTTP. These are added in there. And you also have to have heat-stable DNA polymerase. Okay, these are all added into the reaction. And the process generally involves 3 steps. So the steps that we go through, the first step you have is a strand separation. Strand separation is going to be followed by hybridization of primers. And then we have DNA synthesis that occurs. Strand separation will occur by heating the solution to 95 degrees Celsius for about 15 seconds. So we heat to 95 degrees Celsius for 15 seconds. It's a brief period that we heat that to, and that allows for the melting of those strands. Then hybridization is going to occur when that solution is abruptly cooled, and it's going to be cooled to 54 degrees Celsius to allow the primers to hybridize to a DNA strand. Those primers are typically 20 to 30 nucleotides in length. Then our DNA synthesis is going to occur when the solution is once again heated, not quite as high. It's going to be heated to 72 degrees Celsius, which is the optimal temperature for that heat-stable polymerase. And this 3-step process represents 1 cycle of the PCR. So this process is carried out many times, which we're showing over here on the right. It's carried out many times depending on how amplified we need that DNA to be. So again, I do have a video or a hyperlink here for a video that will show you a little bit more detail. Now, it does use slightly different terminology, but the overview of the technique is the same. So I'll add that hyperlink to the course shell for easier access. In terms of features of PCR, when we're using PCR, the sequence of the target doesn't need to be known. We don't need to know what piece we're looking for. The target can be larger than the primers that we're putting in. The primers don't need to be perfectly matched to the flanking sequence. PCR itself is highly specific and it's very sensitive. So this is a very useful technique, commonly used technique. Now, another way we can look at DNA is we can form recombinant DNA. And there's a method that's called the cohesive end method that allows us to do this. So in order to allow for manipulation of recombinant DNA, we actually need a vector. So a vector is going to be necessary. And there are different types of vectors that exist, but kind of the 2 main choices for vectors that we will encounter are plasmids and bacteriophage lambda. Okay, these are common choices or choice vectors, especially when dealing with E. coli cloning. Now, plasmids, these are circular DNA, and that serves as an accessory chromosome in bacteria. The lambda phage is a virus. So plasmids are circular DNA, and the bacteriophage, that would be a virus. So the vector that we have to take that vector and put the DNA of interest into it. So first we have to prepare the vector, and it's prepared to accept DNA by cutting with restriction enzymes. So we add restriction enzymes. They're going to come in and cut based on whatever particular sequence they're looking at. And it's going to result in staggered cuts. And these staggered cuts will produce complementary single-stranded ends, which we call cohesive ends. And that cohesive ends basically just comes from the affinity of the strands and the affinity that they have for one another. So any DNA fragment with the same cohesive end can then be inserted into the vector. Once we take that insertion and put it into the vector, then DNA ligase can join and anneal the strands together. So you take your vector and you cut it, and you take your sample and you cut it, and it should form complementary strands that will allow us in the end to mix these things together. So this is showing the process of forming these cohesive ends. There are situations where the target DNA doesn't have the proper restriction sites to produce the appropriate cohesive ends, or the ends that we need to stick it into the vector. If that's the case, we can still use this method. However, because short chemically synthesized linkers are available, we can use those in order to add to them and create the sticky ends or the cohesive ends. So we can essentially add the appropriate portions to virtually any DNA molecule so that we can cut it with restriction enzymes and get it into our vector. So we said when we use these vectors, we have two different types, and the first is the plasmid. So this is showing an example of the plasmid. In this particular example, DNA can be inserted into the polylinker region, which is shown here, that polylinker region. The insertion of the DNA will disrupt the natural process. In this case, so this particular plasmid actually is a LAC-Z alpha gene, which encodes ultimately for the production of beta galactosidase. And that's an enzyme that allows for the cleavage of galactose, which is milk sugar. And so when we insert the DNA into this, we're changing the general structure of this particular plasmid, and it's going to disrupt that natural process. So if the enzyme is produced, it will act as a synthetic substrate to release a blue dye. So this is kind of the whole experiment that we're looking at here. If the DNA is altered and recombinant DNA is added, then the protein is not produced and we will not see a blue dye in our sample. So those bacteria which have the insert can be identified based on the absence of the blue dye. So in this particular experiment, we have our plasmid. We have the substance added in there. So for some of these plasmids, we're going to have the DNA inserted into that vector, and we are going to end up seeing blue dye. So the normal process will be these blue spots here. These have not been altered. But then we do see some that have the white spots, and those are going to represent the altered version of this. So we've modified that overall vector. So then we said that the other type of host that we see commonly, or the other type rather of vector that we see commonly, is the lambda-phage. Now lambda-phages can choose to either destroy the host or to become part of the host. And so that depends on the different pathway that it takes. That's that lytic or lysogenic pathway of viruses. So remember that lambda-phages are viruses. So if it becomes part of the host, we call this the lysogenic pathway. So you have your lambda-phage, which comes in and it is going to essentially infect that bacterial cell. Okay, and it's going to take its genetic information and it's going to be inserted in here. And then the lysogenic pathway we see here, that insert is going to become part of the host. And then that is going to be replicated for many generations. Okay, so it's possible that that new DNA is replicated. But in this particular case, it's inactive. Okay, so the lysogenic pathway, it's coming in here and it's integrating into that DNA structure. The other pathway is the lytic pathway. And in that case, so the genetic information of that virus comes in and it is going to replicate within there without inserting into the host DNA. And once that replicates, it actually results in destruction of the host. And when that happens, you have all these new little viruses that are going to be released and can then go on to attack other cells. Okay, so if we take that lysogenic pathway and it's inactive and it's not killing the cell, we can have some environmental changes that can trigger the expression of that dormant viral DNA. So over time, the expression will lead to progeny DNA and eventually lysis. So in other words, lysogenic pathways at some point can and will be converted to lytic pathways. The phage that we have can be modified. So we can modify the genetic information that's coming in from that particular phage. And we do that by splicing it with foreign DNA, much like we did the plasmid. So this is just showing if we're taking that lambda DNA that's part of that virus, we can alter it. So here we're altering the information, package it back into that virus, and then that will then go into the host substance. So one of the things that we can do with this technology is we can create genomic libraries. So complex genome is going to be fragmented. Different fragments are inserted into different vectors. Here we're using lambda phages in order to do that. And then once we do that and we modify, the individual portions that we've created can be amplified and identified. So this is just looking at kind of these genomic libraries or these pieces, and you have fragmentation that puts them into that lambda DNA. So we're taking these now substances that are in the lambda, creating these now viruses which can go on, and they serve as our phages just depending on what we need them for. Now we can screen for genomic libraries, and the screening process allows you to find the gene of interest among all of the other genes that you have. Usually this involves plating the recombinant phages on bacteria. Infected bacteria will result in a plaque formation on a nitrocellulose plate. A replica will be made of that nitrocellulose. The infected bacteria and phage DNA are released by the lyse cells. They stick to that sheet, and then the sheets are treated and labeled and then analyzed for spots of interest, which would contain the gene of interest. So we have our plaque that forms. You transfer onto your nitrocellulose sheet. You add some probes in there, and the particular portion of interest, because of that probe or that label you put in there, when you analyze it, you're able to detect where it is. And in this case, it's a radioactive version, which we're picking up on X-rays. So what kinds of things can we do with this? Well, some of the implications of this technology, for example, the production of proinsulin. So people who are diabetic will benefit greatly from this technology. So you can create proteins that have new functions. So here the proinsulin is actually a precursor of insulin. We can synthesize that by using genetically altered clones of E. coli. The clones can contain the mammalian proinsulin gene, and then we can end up using that for medicinal purposes. Now, one last thing that the chapter addresses, or that we'll address related to the chapter, are different types of mutations that can be introduced or made. So the recombinant DNA allows for specific mutations to be made. We are intentionally mutating DNA. Generally speaking, mutations come in the form of deletions, insertions, and substitutions. Some of the types of processes that we see for these mutations include the site-directed mutagenesis, cassette mutagenesis, and mutagenesis by PCR. Now in the site-directed, this allows for the generation of mutant proteins with a single amino acid substitute. So this is going to be our substitution is going to be the case there. Cassette mutagenesis allows for more complex sets of mutations to take place. And the mutagenesis by PCR uses primers to introduce insertions, deletions, and substitutions. So mutagenesis by PCR will allow us to kind of do all of these things. So all of these methods combined allow for the development of designer proteins, or proteins that are not necessarily found in nature. So this chapter presents information that has led to significant advances in biotechnology, especially as it relates to DNA technology and understanding the genome.